Hello student, now we study about properties of good or ideal cloning vector for rDNA technology, that is recombinant DNA technology. So properties or characteristic of ideal vector. The first property, vector should be able to easily transferred into host cell. Second property, it should be able to replicate independently through ORI gene, which is also called replication origin, so that as vector replicate, multiply copies of DNA insert are also produced. Third properties of ideal cloning vector, that is it should have marker genes for antibiotic resistance and restriction enzyme recognition site within them. Fourth property, it should have suitable control elements like promoter, operator, ribosomal binding site. Next question, write short note on constructed plasmid. So definition of constructed plasmid. The plasmid obtained naturally do not possess all characteristics which are present in ideal cloning vector. So they are constructed by inserting genes for antibiotic resistance. So example of constructed plasmid, these are PBR 322, PBR 320, PACYC 177 are example of constructed plasmid. Out of these, PBR 322 is mostly used in rDNA technology, that is recombinant DNA technology in plants. Next question, write short note on example of vector, that is plasmid and plasmid for plants. So first short note about plasmid. The plasmid most commonly used in recombinant DNA technology are those that replicate in E. coli, that is Escherichia coli. Investors have engineered these plasmid to optimize their use as vector in DNA cloning. So this is diagram for plasmid cloning vector which show ORI. ORI means replication origin. AMP, that is drug resistance gene. These are the region into which the foreign DNA can be inserted. So this is diagram for plasmid cloning vector. The next one, that is vector, plasmid vector for plant. So important vector for carrying new DNA into many type of plants is plasmid that found in Agrobacterium tuminificans. This bacterium live in soil, cause plant disease called as crown gall, which is characteristic by presence of overgrowth or tumor in plants. So Agrobacterium contain plasmid called as Ti, that is for tumor inducing. The plasmid, that is Ti plasmid, contain transpond Transpond means jumping gene called tDNA, which insert copies of itself into chromosome of infected plant cell. Number 5th character, the transpond with new DNA can still inserted into host cell chromosome. The last characteristic of plasmid vector for plants, that is a plant cell contain this DNA can then be grown in culture or induced to form new transgenic plant. So these are different properties of ideal or good cloning vector, as well as the characteristic of plasmid and plasmid for plants.